cell cycle is divided into a number of parts, the longest being the interphase, which is divided into 3 subsections, including G1, S, and G2. In G1, the cell synthesizes proteins that are needed for the S phase, which is mainly used for DNA replication. It also replicates organelles and grows in size. In the S, or synthesis phase, DNA replication occurs and all genetic material is copied. This is the longest part of the cell cycle because it is crucial to carefully revise the copied DNA for mistakes. In G2, the cell continues to grow and prepares for mitosis. After interphase is the mitotic phase, which is divided into mitosis and cytokinesis. Mitosis, which is the division of the genetic material in the cell, takes place in 4 stages: prophase, metaphase, anaphase, and telophase. After telophase, cytokinesis occurs, which is the actual splitting of the cell. Sometimes cells leave the cell cycle and enter the G0 phase, or resting phase, in which the cell is neither dividing nor preparing to divide. Some cells, such as neurons and heart muscle cells, enter this phase once they reach maturity.
Mitosis is the process of cellular division that produces 2 genetically identical cells. Its main purposes are growth, maintenance, and repair. The 2 cells produced are diploid cells. They contain 2 N chromosomes, which is the full genome. Mitosis is divided into 4 stages: prophase, metaphase, anaphase, and telophase. In prophase, chromatin begins to condense. The nucleolus disappears and centrioles move towards opposite ends of the cell. Then the nuclear membrane dissolves. Proteins form kinetochores on chromosomes and microtubules attach to the kinetochores, moving the chromosomes. In metaphase, microtubules align the chromosomes along the middle of the cell nucleus. This line is the metaphase plate. This helps ensure that when the chromosomes are separated, each new nucleus will receive 1 copy of each chromosome. In anaphase, sister chromatids separate at the kinetochores and move to opposite sides of the cell. As soon as the chromatids are split, the number of chromosomes in the cell has doubled. The movement of chromosomes results from a combination of degradation of the kinetochore microtubules and elongation of non-kinetochore microtubules. In telophase, chromatids arrive at opposite sides of the cell and new nuclear membranes form. The chromosomes uncoil into chromatin form and are no longer visible under a light microscope. Microtubules also disperse. Cytokinesis is when the plasma membrane of the cell splits so that the new daughter cell can break off from the parent cell. In animal cells, cytokinesis results when a ring composed of a protein called actin around the center of the cell contracts, pinching the cell into 2 daughter cells, each with 1 nucleus. In plant cells, a cell plate is formed between the 2 cells due to the presence of a cell wall and not just a plasma membrane.
Meiosis is the process of cellular division that produces 4 genetically unique cells. These cells are gametes. The 4 cells produced are haploid cells, and they contain N chromosomes, which is half of the genome. Meiosis is divided into 2 major stages, meiosis I and meiosis II. The purpose of meiosis I is to separate homologous chromosomes. Prophase I is the longest stage of meiosis. Similar to prophase and mitosis, chromatin condenses into chromosomes and the nucleus goes away. Centrioles move towards opposite ends of the cell. However, in meiosis, this stage is where genetic variation occurs. Homologous chromosomes form tetrads and overlap at locations called chiasmata. The exchange of genetic information is called crossing over. In metaphase I, homologous chromosomes line up along the metaphase plate with the help of microtubules. In anaphase I, homologous chromosomes are separated and move towards opposite ends of the cell. In telophase I, chromosomes arrive at opposite ends of the cell and form new nuclei around them. Chromosomes uncoil back into chromatin.
Then the cell undergoes cytokinesis and there are 2 cells each with half the number of chromosomes. The first round of division is now complete. Meiosis II's purpose is to separate sister chromatids. In prophase II, chromatin condenses into chromosome form again and the nucleus disappears. Mitotic spindle begins to form as centrioles move to opposite ends of the cell. In metaphase II, sister chromatins line up along the metaphase plate. In anaphase II, sister chromatids separate and move towards opposite ends of the cell. In telophase II, new nuclei form around the chromosomes. The 4 daughter cells are haploid. Then each cell undergoes cytokinesis again and there are 4 cells, each holding a unique set of genetic information. Meiosis is now complete.